Hi all, this is Sree Balaji, working as a tutor, Department of Physiology, Sree Balaji Medical College and Hospital. Today we are going to see an important topic in general physiology, active transport. In previous class, we all would have come across the transport across cell membrane and one of the classification of transport across cell membrane, passive transport. Today we are going to see about active transport. So learning objectives of today's session is, we are going to see the definition of active transport, classification of active transport, that is primary active transport and secondary active transport. We are going to see the mechanism of sodium potassium pump, and finally we are going to see about the applied aspects. Before we understand active transport, let us briefly see how substances move across the cell membrane. The cell membrane is selectively permeable. It allows certain substances to pass while restricting others. For the cell to survive, nutrients must enter, waste must leave, and ions must be properly distributed. Transport across the cell membrane is broadly classified into three types. The first is passive transport, active transport, and vesicular transport. Passive transport, here what happens is substances move from higher concentration to lower concentration along the gradient without using energy. Examples include simple diffusion, facilitated diffusion, and osmosis. Active transport, in this process, substances move against the concentration gradient, that is from lower concentration to higher concentration. This requires energy in the form of ATP. Example, sodium potassium pump. Vesicular transport, here larger molecules move in or out of the cell by forming vesicles. Here examples are endocytosis and exocytosis. So, transport across the cell membrane ensures proper nutrient supply, waste removal, maintenance of ionic balance, and overall cellular homeostasis. Let's go into the session. Today's session is about active transport. If a substance naturally moves from higher concentration to lower concentration, what happens if the cell wants to move it in the opposite direction, from lower concentration to higher concentration? That movement is not spontaneous. It requires energy, and that process is called as active transport. Definition of active transport. Active transport is the movement of substances across a cell membrane against their concentration gradient or electrochemical gradient using energy in the form of ATP. So basically, it requires energy in the form of ATP. It requires carrier proteins, that is pumps, transport against concentration gradient, also called as electrical gradient or pressure gradient. There are four keywords you must remember, that is against concentration gradient, energy dependent, carrier mediated, and it should be specific. If these four features are present, it is active transport. Since the transport of substances occur against a chemico-electrical gradient, this process is also called as uphill movement. Substances transported actively across the cell membrane, which includes ionic substances and non-ionic substances. Ionic substances such as sodium, potassium, calcium, and non-ionic substances like glucose, amino acids, and urea. Why do cells need active transport? Cells are not just bags of fluid. They maintain a very specific internal environment. For example, sodium concentration is high outside the cell and potassium concentration is high inside the cell. If only diffusion happened, these concentration would equalize. But they do not, because active transport maintains them. Now let us move to a very important concept in active transport, that is carrier protein. We all have discussed that active transport cannot occur without specific carrier proteins. These proteins are embedded in the cell membrane and they help in moving substances across the membrane. Carrier proteins involved in active transport are of mainly two types. One is uniport, symport, or antiport. Let us understand each one clearly. First we'll see about uniport. What is uniport? The word uni means same. So uniport is a carrier protein that transports only one substance in a single direction across the membrane. It carries only one type of molecule in one direction, that is why it is called as uniport. For example, if a carrier protein transports only calcium ions from inside the cell to outside without transporting anything else along with it, this is called as uniport. So remember, one substance, one direction. Now we are going to see about symport and antiport. Now let us discuss the second type. This is slightly more interesting. These carrier proteins transport two substances at a time, and based on the direction in which they move, they are classified into symport, antiport. First we'll see about symport. The word symport means same. So in symport, two different substances are transported in the same direction across the membrane. Both move together. Antiport is, it is in the opposite direction. Here, two different substances are transported in the opposite direction. One substance enters into the cell, others leave the cell. That is why it is called as antiport. So examples are sodium calcium exchanger. Here, sodium enters the cell and calcium leaves the cell. It is in opposite direction, so it is antiport. Now let us understand the mechanism of active transport. We'll see step by step. Step 1 is binding of the substance. First, imagine a substance that needs to be transported across the cell membrane. When this substance comes close to the outer surface of the cell membrane, it does not simply pass through. Instead, it binds to a specific carrier protein present in the membrane. This carrier protein has a special binding site designed only for that particular substance. So the substance attached to the carrier protein. Now what is formed? A substance protein complex is formed. Now we are move on to the step 2. Here formation of substance protein complex is going to happen. Once the substance binds, the carrier protein undergoes a structural change. This change in shape is very important. Why? Because it allows the complex to move across the cell membrane. In active transport, this change usually requires energy from ATP. Now we are going to see about the step 3, movement across the cell membrane. After the change, the substance protein complex moves towards the inner surface of the cell membrane. You can imagine it like a revolving door. The carrier protein changes its orientation from facing outside to facing inside. Now we are going to see about the step 4, which is releasing of the substance. Once it reaches the inner surface, the substance is released into the cell. Why does it release? Because the affinity of the carrier protein changes after the change. So the substance detaches and enters the cytoplasm of the cell. Now step 5 is carrier protein returns to its original position. So now the carrier protein is empty. It does not stay inside. It again undergoes another shape change and moves back to its original position on the outer surface of the membrane. Now it is ready to transport another molecule, and this cycle continues again and again. Now we are going to see about the types of active transport. The first type is primary active transport and the second type is secondary active transport. First we'll see about the primary active transport. It is defined as the movement of substance across the cell membrane against a concentration gradient by directly using energy derived from ATP hydrolysis. By this method, the substances like sodium, potassium, calcium, hydrogen and chloride are transported across the cell membrane. In primary active transport, the ATP is directly broken down, energy is immediately used, the transport protein itself acts as an enzyme. Why it is needed? In the body, many ions need to be maintained at unequal concentration inside and outside the cell. For example, sodium is high outside the cell and potassium is high inside the cell. Calcium is very low inside the cell. Hydrogen ions are secreted into the stomach. These gradients cannot be maintained by diffusion. They require energy, and that is where primary active transport comes in. Primary active transport of sodium and potassium. We are going to see about the sodium-potassium pump elaborately. Sodium and potassium ions are transported across the cell membrane by means of a common carrier protein called as sodium-potassium pump. It is also called as sodium-potassium ATPase pump or sodium-potassium ATPase. The structure of the sodium-potassium pump is, it consists of two main subunits. First is alpha subunit and beta subunit. The alpha subunit acts as the functional unit and the beta subunit acts as the supporting unit. Alpha subunit is the largest and most important subunit. It performs the actual pumping action, and functions of alpha subunit is, it binds sodium ions inside the cell and potassium ions outside the cell. It also binds ATP, has ATPase activity, gets phosphorylated, undergoes conformational change. Beta subunits are giving structural support. The beta subunit is smaller. It stabilizes the alpha subunit, helps in proper folding, helps transport the pump of the membrane, maintains structural integrity. It does not directly pump ions. Now we are going to see about the mechanism of sodium-potassium pump. With this picture, we'll discuss the mechanism of sodium-potassium pump. Initially, the pump is open towards the inside of the cell. In this position, it has high affinity for the sodium. 3 sodium ions from the cytoplasm binds to the pump. So 3 sodium attaches to their binding site. Now ATP binds to the pump on the cytoplasmic site. ATP is broken down into ADP and phosphate and energy. The phosphate group attaches to the pump. This provides the energy required for the next step. Because of phosphorylation, the pump changes its shape. Now it becomes open towards the outside of the cell. At this point, its affinity for sodium is going to get decreases. Since affinity decreases, the 3 sodium ions are released outside the cell. Now sodium concentration remains high outside. In this outward facing position, the pump now has high affinity for potassium. 2 potassium ions from outside bind to the pump. After potassium binds, the phosphate group detaches from the pump. This triggers another shape change. The pump returns to its original inward facing position. Now affinity for potassium decreases. The 2 potassium ions are released inside the cell. Now the potassium is ready to start the next cycle. The sodium-potassium pump works like a two-sided gate. First it opens inside, grabs sodium, then closes and opens outside to release it. Then it grabs potassium from outside and opens inside. Again it is going to release it. Now we are going to see about the functions of sodium-potassium pump. It helps in maintenance of resting membrane potential, maintenance of intracellular ion concentration, regulation of cell volume, provides sodium gradient for secondary active transport, role in muscle function, energy consumption and metabolic role. Now we are going to see about the transport of calcium ion by using calcium pump. This pump uses a specific carrier protein. It requires ATP for energy, works through the enzyme activity of ATPase. The calcium pump maintains very low intracellular calcium level, which is extremely important because calcium acts as a signaling molecule inside the cell. It is also present in sarcoplasmic reticulum of the muscle cell, where the calcium is pumped inside to help in muscle relaxation. In mitochondria, where calcium is stored and regulated. Now we are going to see about the transport of hydrogen ion by using hydrogen pump. So this pump uses a specific carrier protein. It requires ATP for energy, functions through enzyme activity of ATPase. Hydrogen pump have important role in 2 major organs. One is stomach, another one in kidney. In the parietal cells of the gastric glands, hydrogen pumps help in the formation of HCl. This acid is essential for the digestion and activation of enzymes like pepsin. In kidney, the distal convoluted tubule and collecting duct, hydrogen pumps secrete hydrogen ion from the blood into the urine. This helps in maintaining acid-base balance of the body. Now we are going to see about the second classification, which is nothing but secondary active transport. Now this is slightly different. Here ATP is not directly used. Instead, energy is derived from stored gradient created by primary active transport. Most commonly, sodium gradient created by sodium-potassium pump. Secondary active transport is the movement of substance against the gradient using energy stored in the ionic gradient. Types is co-transport and counter-transport. Sodium co-transport, this is also called as sodium symport. We have discussed about the symport, uniport and antiport in the before slides itself. In this mechanism, sodium moves along with another substance through the same carrier protein. Sodium co-transport is a process in which sodium moves down its concentration gradient. Another substance moves against its gradient. Both move together in the same direction through a carrier protein called as symport. The sodium gradient is created by the sodium-potassium pump using ATP. So ATP is used indirectly. When sodium moves into the cell, down its gradient, energy is going to get released. That released energy is used to transport another substance into the cell. So this is a perfect example for the secondary active transport. Substances commonly transported along with sodium include glucose, amino acid, chloride, iron, iodine and urate. All these use sodium gradient for their absorption and reabsorption. The carrier protein involved in sodium co-transport has 2 receptor sites in the outer surface of the membrane. Among these 2 sites, one site is for sodium and the other site is for the second substance like glucose or amino acid. Both must bind for the transporter to function. Sodium co-transport of glucose. Now let us understand the example glucose transport. This occurs mainly in small intestine, renal proximal tubule during reabsorption. In the small intestine, it occurs during absorption. Step-by-step mechanism. Once sodium ion from the extracellular fluid binds to its receptor site, one glucose molecule binds to its receptor site. When both are bound, the carrier protein becomes activated. It undergoes conformational change. Both sodium and glucose are transported into the cell together. So sodium moves down its gradient and glucose moves against its gradients. This transport is called as sodium-glucose linked transporter. Now we are going to see about the sodium co-transport of amino acid. The transport of amino acid is slightly more complex. Carrier proteins for amino acids are different from glucose transporters. It is more specialized. There are about 5 different sets of carrier protein. Each one transports specific types of amino acids depending on molecular weight, chemical structure. This transport also occurs in small intestine for absorption, renal tubules during reabsorption. Now we are going to see about the sodium counter transport, that is antipode system. Sodium counter transport is a type of secondary active transport in which a substance moves across the cell membrane in exchange for sodium ion. It is called as antipode because two substances moves in opposite direction. Movement occurs through a carrier protein. Major sodium counter transport systems are sodium calcium counter transport, sodium hydrogen counter transport. First we'll see about sodium calcium counter transport. Sodium moves into the cell, calcium moves out of the cell. Both moves in the opposite direction through a carrier protein. It present in many cells, especially important cardiac muscle cell for regulating intracellular calcium level. Sodium hydrogen counter transport, it occurs mainly in renal tubular cell. Sodium moves from the tubular lumen into the tubular cell. Hydrogen ion moves from the tubular cell into the lumen. It is important for acid-base balance in the body. Applied physiologies of today's session. First applied physiology is abnormality of sodium-potassium pump. The sodium-potassium pump is essential for maintaining resting membrane potential and ionic balance. Any reduction in its number or function can lead to a disease. The clinical importance are cardiac failure, hypertension. Cardiac failure, a reduction in the number of activity of sodium-potassium pump in the myocardium is associated with cardiac failure because ionic imbalance affects cardiac contractility. Hypertension, excess reabsorption of sodium in the renal tubule increases blood volume leading to hypertension. Next, channelopathies, ion channel diseases. Channelopathies are disorders caused by mutation in the genes encoding ion channels. So, sodium channel disease, dysfunction can cause muscle spasm. It will also lead to a syndrome called as Liddle syndrome. Next, potassium channel disease can cause cardiac arrhythmia, neonatal epileptic seizure. Next, we are going to see about chloride channel disease. It can lead to renal stone formation and cystic fibrosis. To summarize, in today's session, we have seen about the definition of active transport, classification of active transport, that is primary active transport and secondary active transport. Under primary active transport, we have seen elaborately about sodium-potassium pump and functions of sodium-potassium pump. Under secondary active transport, we have come across co-transport and counter-transport, and finally, we all have come across the applied aspects of today's session. Thank you for your patience listening.